Hello everyone. Today we are going to study cell cycle and cell division. So before begin the lecture, I would like to ask you a question. How does our any of the cell damage or injury get recovered? How a single cell or a zygote converted into a baby? So all the answer of these questions lie in this chapter, which is cell cycle and cell division. So basically in the cell cycle, we are going to study what is cell cycle, what is cell division, what are the types of cell division, what are the significance of cell division, and many more things. So let's begin with the topic cell cycle. So what is cell cycle? Cell cycle is a sequence of events in which cell grows, replicates its DNA and divide into a daughter cells. This is called cell cycle. Cell cycle is an event of a complete life cycle of a cell. So it is a sequence of event. We will learn everything by breaking down into small points so that we can remember each and every point very clearly. It is a sequence of events. What are the events in the cell cycle? Cell growth. Second is DNA replication. Third is cell replication. And third is division. Division, basically cell division. So when does growth occur in the cell? Growth of a cell, it is a continuous process which continues throughout the cell cycle. It means सेल ग्रोथ कब करता है? तो सेल का जो ग्रोथ है वो कंप्लीट लाइफ साइकिल में, इट मीन जितना भी उसका लाइफ है, सेल की लाइफ जितनी भी है उसमें उसका कंटिन्यूज रहता है. इट मीन्स वो अपने आप का अपना साइज इंक्रीज करता है, जो भी डैमेज है उन सब चीज़ों को रिपेयर करता है, प्रोटीन सिंथेसिस करते रहता है. ऑल दीज आर under the growth. It is a continuous process in which what happens? Increase in the size, change in the shape, protein synthesis, transcription, translation, all occurs in the cell growth. And what is DNA replication? In order to divide into two equal daughter cells, the DNA, which is the genetic material of the cell, has to be divided into two equal parts. So DNA replication is required. What happens in DNA replication? In DNA replication, one copy of a DNA synthesizes the exactly same copy of a DNA, so that one DNA can go to one of the daughter cell and another part can go to the another daughter cell. So one copy of a DNA, genetic material, synthesizes another strand of a exactly similar same DNA strand in order to get divide equally into the daughter cells. What is a cell division? Now, cell division is the last step in which a fully growth matured cell divide into two equal halves and multiplies its number. By dividing a cell divide itself to multiply their number, it means increase their number. Now we will see the different events of cell cycle. Firstly, in the cell cycle, there is two phases. One is growth phase, or in the terms of cell cycle, we called it as interphase. And another is M phase. So what happens in the interphase? Interphase is also known as resting phase. Resting phase, or we called it as preparatory phase also. So what happens in the interphase, resting phase or preparatory phase? In the resting phase is it's not mean that in the interphase or resting phase cell rests. In the interphase, cell prepare itself for and growth itself prepare itself for the cell division. In so in the interphase, what happens in the cell? There are uh, three different types of phases. Interphase is divided into three different phases. First is G1 phase, second is synthesis phase, and third is G2 phase. G1 phase means gap phase 1 or growth phase 1. S means synthesis phase. I'll write here gap phase 1, synthesis phase. Third is gap phase 2. So what happened in these three phases of a cell cycle? In the G1 phase, in this G1 phase or a growth phase 1, cell growth occur, increase in the cytoplasm, increase in the cell cycle, increase in the cytoplasm, increase in the cell size, synthesis of some important proteins, transcription and translation occurs in the synthesis. In the synthesis phase, the replication of a DNA occur. That's why we called it synthesis phase. And in the G2 phase, cell again prepare itself, again synthesizes a protein and duplicates its cell organelle for the cell division. So interphase comprises of almost 95% of any cell cycle. It is the more than 95% of a cell cycle. Now in the M phase is the division phase where cell divide itself. So in the M phase, there is two phases. First division occurs. Firstly, the division of a cell starts from the nucleus of the cell. So first phase is known as karyokinesis. What what does karyokinesis mean? The karyokinesis is the division of nucleus, and the other is cytokinesis. So in the karyokinesis, where nucleus divide into two equal half, it is divided into karyokinesis is divided into 4 different phases. Just remember the names of all the phases in here. Later on, we will discuss each and every point or a single detail occurring in the each and every phase. So in the karyokinesis, there is prophase, Pmax. Okay. Pmax is prophase मेटाफेज, एनाफेज, and here telophase. So let's summarize this slide. Cell cycle, we know this definition of a cell cycle. Cell cycle is a sequence of events through which a cell passes through cell growth, cell cell growth, DNA replication, and cell division. These are the three main events occurring in a cell cycle. It means सेल अपने पूरे लाइफ में इन्हीं तीन इवेंट से पास होता है, जो कि पहला है सेल ग्रोथ. इट मीन्स वो अपना जो भी साइटोप्लाज्म कंटेंट है, अपना साइज है, खुद को मेच्योर करना, वो सारा ग्रोथ में आता है. एंड देन इन दिस डीएनए रेप्लिकेशन, जिसमें कि सेल अपने आप को, अपने डीएनए को डुप्लीकेट करता है ताकि वो डॉटर सेल बने, तो उसमें 2 इक्वल हाफ न्यूक्लियस प्रेजेंट हो. और सेल डिवीजन मीन्स वही डिवीजन ऑफ अ न्यूक्लियस एंड द साइटोप्लाज्म, ताकि 2 डॉटर सेल बन सके. तो सेल साइकिल में पहले उसे 2 फेजेस में डिवाइड किया गया है. फर्स्ट इज इंटरफेज एंड अनदर इज एम फेज. एम फेज बेसिकली 5% ऑफ अ कंप्लीट सेल साइकिल. तो सपोज ये एक सेल साइकिल है, तो इसका ये जो 5% पार्ट जो है वो एम फेज है, बाकी का रेस्टिंग फेज विच इज द 9%. इंटरफेज को हम लोग आई फेज भी बोलते हैं. वी डिनोट इट एज आई फेज एंड माइटोटिक फेज मीन्स एम फेज. तो इंटरफेज में अगेन उसको हम लोग 3 पार्ट्स में डिवाइड करते हैं. फर्स्ट इज गैप 1 ग्रोथ 1 फेज, जिसमें वो अपना ग्रोथ करता है. देन सिंथेसिस फेज, जिसमें डीएनए रेप्लिकेशन होता है. एंड देन अगेन थर्ड G2 फेज विच इज ग्रोथ फेज 2. फिर से उसमें वो अब डीएनए खुद को सेल डिवीजन के लिए प्रिपेयर करता है. यहाँ पे सेल खुद को प्रिपेयर करता है क्योंकि वो उस टाइम वो नेसेंट होता है, मीन्स कि वहां पे अभी वो डॉटर सेल बनकर आया है, जिससे कि वो अपना ग्रोथ कर रहा है. अप टू अ सर्टेन लिमिट वो अपना ग्रोथ करता है. फिर डीएनए रेप्लिकेशन होता है एंड देन फिर वो सेल डिवीजन के लिए खुद को प्रिपेयर करता है. नाउ इन द एम फेज मीन्स डिवीजन होना है. तो यहाँ पे पहले तो न्यूक्लियस का डिवीजन होता है एंड देन साइटोप्लाज्म का डिवीजन होता है. तो न्यूक्लियस के डिवीजन को हम लोगों ने 4 पार्ट में फिर से डिवाइड करा है. प्रोफेज, मेटाफेज, एनाफेज एंड टीलोफेज. इन सारे चारों फेजेस में डिफरेंट इवेंट्स होते हैं, डिफरेंट अरेंजमेंट्स होते हैं न्यूक्लियस का और सेल ऑर्गेनेल्स का और सारी चीजों का. तो हम लोग वो सब नेक्स्ट स्लाइड में डिस्कस करेंगे. एंड इन द साइटोकाइनेसिस विच इज डिवीजन ऑफ अ साइटोप्लाज्म. और फिर यहाँ के बाद हम लोगों को डॉटर सेल मिल जाता है. वी गेट डॉटर सेल, 2 डॉटर सेल्स. ओके, नाउ लेट्स begin with the G1 phase. In the G1 phase, growth 1 phase, the first thing which occur in the growth phase 1 is growth in the size. First point is growth in size. It means here single cell increases its size. So if a cell increases its size, what happens? Increase in the cytoplasm. Okay, सेल जब अपने आप को अपना ग्रोथ कर रहा है तो उसमें क्या होगा? बिकॉज डीएनए कंटेंट तो वही रहेगा, न्यूक्लियस जो है वो उसका उतना ही रहेगा. तो इसमें ग्रोथ में क्या होगा? उसका सेल वॉल जो है वो सेल मेम्ब्रेन एक्सपैंड करेगा. इंक्रीज इन साइटोप्लाज्म, इंक्रीज होगा क्या में? उसका सेल के साइटोप्लाज्म में, ताकि वो बड़ा हो सके. देन और क्या होगा? ट्रांसलेशन एंड ट्रांसक्रिप्शन. We called it as protein synthesis. Protein synthesis means transcription and translation. Here we write translation plus transcription. Transcription is the formation of a RNA from DNA molecule, and translation is the formation of a protein from a RNA molecule. So we can write translation as a protein synthesis, and we can also write transcription as a RNA synthesis, which also occur in this phase. Protein, what kind of proteins will synthesize here? किस तरीके के प्रोटीन यहाँ पे सिंथेसाइज होंगे? तो as we know कि जीवन फेज के बाद एस फेज आने वाला है, जिसमें कि डीएनए रेप्लिकेशन होना है. तो जब डीएनए रेप्लिकेशन होना है तो डीएनए के बाइंडिंग के लिए हमें एक काइंड ऑफ प्रोटीन चाहिए, रिक्वायर्ड होता है, जो कि होता है हिस्टोन प्रोटीन. तो यहाँ पे सिंथेसिस हो सकता है हिस्टोन प्रोटीन का, ताकि सेल जब एस फेज में जाए तो वहां पे उसे वो हिस्टोन प्रोटीन अवेलेबल हो, ताकि रेप्लिकेशन हो सके. We can also write here increase in the cytoplasm and cell membrane. Cell membrane भी expand करेगा. Now G1 phase. Now here is a note that G1 phase is the deciding phase whether cell will divide or not. G1 phase is a deciding phase whether cell will divide or not. If cell will divide, then it will prepare itself for replication process or all the other processes. And if cell will not divide, it will, if not, then cell will enter into G0 phase. G0 phase is the resting phase. This is the resting phase, means cell here we will not call it as resting phase. Cell here enters into cell here get differentiated. It means cell is get differentiated to perform any specific function. So for example, RBC, we all know that the life cycle of a RBC, lifetime span for RBC is 4 months. So when the RBC get mature, it enters into the G0 phase, where the cell get differentiated to perform specific function. In the G0 phase, cell remain metabolically active, but it do not in the G0 phase, cell remain metabolically active but do not participate into cell division. When the cell came back from the G0 phase and again enters into the G1 phase and prepare itself for the cell division. But the example of which the cell which remain in G0 phase is neurons, RBC, heart cells. These are the example of cells which remain in G0 phase. Okay. So here the G0 phase enters ends. G1 phase is the longest phase. It is the longest phase of interphase, longest phase of I phase. It is basically 10 to 11 hours. For example, a human cell can, requires 24 hour. Human cell require 24 hours for the complete to complete a cell cycle. Out of 24 hours, cell takes 10 to 11 hours of 24 hours to get complete the cell cycle. Now here G1 phase end. We will next move towards the S phase, synthesis phase. Now let's summarize this slide. After the formation of a two daughter cells, cell G enters into the G1 phase, where the growth of a cell occur, means increase in the size, increase in the cytoplasm, expansion of the cell membrane, protein synthesis, RNA synthesis. It is the longest phase of an interphase. It takes 10 to 11 hours to complete the G1 phase. G1 phase is the deciding phase whether the cell will divide or not. Now we will move toward the next phase, which is our synthesis phase. Now synthesis phase and S phase simply indicate the replication of a DNA or a duplication of a genetic material. The main function which is performed by the cell in the S phase is the replication of a DNA. Replication of DNA. What happens in the replication of a DNA? We know DNA is a double helical structure. It is a double helical structure. What happens in this? This both the strand get separate like this at a portion only. Not complete DNA get open at the same time. Only a few portion or a part of a DNA get unwind, and it synthesizes a new strand of a DNA, so that by the end of the complete replication we get two different strands of DNA. In the replication of a DNA means duplication of a DNA, means from the one DNA strand we get the another same new DNA strand. That's it. And here we will one more very important event occur in this synthesis phase, which is duplication of a centriole. What is centriole? Centriole is a cytoskeleton. Centriole is a cytoskeleton which help in the cell division, as the centriole is the site from where centriole वो जगह है जहां centriole वो organelle है जहां से spindle fibers rays निकलते हैं and get attaches to the chromosome to divide into two. Centriole of a duplication of a centriole is required. Why? Because two centriole will duplicate and form two centriole, so that both the centriole can move to the opposite poles and divide this genetic material into two different parts. That's why the duplication of a centriole is required. Why duplication of a DNA required? Because duplication of a DNA required because it is the genetic material, and it is the genetic material of a cell, and both the daughter cell can get equal amount of a genetic material or equal amount of a DNA, and duplication of a centriole is required to divide the cell into two equal halves. Here the production of histone protein also occur. Now we will move towards the next phase, which is G2 phase. What are the events happens in the G2 phase? Which is growth continues here, means cell continue to grow. Synthesis of some cytoskeleton, or we can say synthesis of tubulin protein. What is tubulin protein? Spindle fiber. Spindle fiber is a kind of a cytoskeleton which is made up of tubulin protein. So, synthesis of tubulin protein is very important or a very required process in the G2 phase, as because after the G2 phase, cell will enter into the M phase where cell has to divide into two equal halves by the help of a spindle fiber. And increase in the number of mitochondria. Mitochondria increases. Golgi bodies. Golgi bodies also increases here. Now here our interphase ends, and next we will move. The synthesis phase continues for 6 to 8 hours, and this phase also continues for 7 to 8 hours. This is the S phase. S phase is the shortest phase of an interphase, and it continues for 6 to 8 hours. 6 to 8 hours, whereas the G2 phase, it continues for about 8 to 10 hours. Not 10 hours, basically 7 to 8. 7 to 8 hours this, and this is for 5 to 6 hours. Here the G2 phase ends. Now next we will move towards the M phase. Before moving towards the M phase, let us discuss some the type of a cell division, so that we can differentiate between the difference in the cell cycle of both the cell type and cell division type. There are cell division. There are two types of a cell division. First is mitotic. This is the very important topic, so please pay attention here so that we can understand it very clearly. First is mitotic division, and sorry, mitosis, not mitotic. First is mitosis, another is meiosis. So in mitosis is also known as equational division. I will discuss why it is called as equational division and it is called as reductional division. In the mitosis, one single cell divide into two equal half to give two daughter cells. This cell is known as parent cell and these two cells are known as daughter cell. Here, single cell divide into exactly two equal half to give daughter cells. In the mitosis, the parent cell and the daughter cell both are exactly similar to their parent. It means the daughter cell है हमारा वो एकदम exact same होगा. Each and everything is exactly same as the parent cell. All we know the ploidy of the human cell, which is a diploid, means here the parent cell is diploid. There is 2n. Then the daughter cell which will form is also 2n. It means it has two sets of chromosome. Now in the meiosis, why it is called as reductional division? That's why we called it as equational division, because both the daughter cell the parent cell is dividing into equal half to produce two similar same daughter cell. That's why we called it as equational. Equation equal division. In the reductional division, reduction means to reduce हो रहा है इसमें जो सेल की जो प्लॉइडी है वो रिड्यूस हो रही है. लेट्स सी हाउ. Here is the parent cell. Here it is parent cell where whose ploidy is 2n. It will divide into two cells firstly, where the ploidy of a cell became n, it is haploid, and then again these cells divide into two to produce four daughter cells. Here only two daughter cells are produced. डॉटर सेल जो है वो कितनी हो रही है हमारी 2, और यहाँ पे डॉटर सेल्स हमें कितने मिल रहे हैं? We are getting 4 daughter cells here and we are getting 2 daughter cells in the mitosis. In the meiosis we get the 4 daughter cells. So what is the difference of in between these daughter cells and these daughter cells? The difference in the daughter cell is here the ploidy is half but the content of a DNA is double, whereas here the content of a DNA is single. Why it is called as reductional division? Because the ploidy of a cell is getting half. That's why we called it as reductional division. These are the two kinds of cell division which is occurring in all the in living organisms. Mitosis occurs in somatic cells. For example, liver cell, RBC, skin cells. Skin cell means in our skin. What uh, epithelial cells or any many cells. There are so many cells. Or in meiosis occurs in the gamete formation only, sex cells only. Gamete formation. We will discuss the significance of meiosis after studying the different phases of meiosis. Let me explain in a just one or two lines that why meiosis is required. Meiosis is required to maintain the ploidy in a zygote, which will result into the a newborn baby. So in the zygote, what happens? The two different gametes are fused together to form one single zygote. So the both the gamete must carry only one set of chromosome, and both the only one set of chromosome, so that the one zygote can get one set of chromosome from the male gamete and one set of a chromosome from female gamete, and both this is the set of a chromosome which is from this is the male gamete which carry one set of a chromosome, and this is the female gamete which also carry one set only one set of a chromosome, and this and these both gamete fuse together so that it will form a one complete pair of any chromosome. That's why the reductional division is very very important in the gamete formation to maintain the ploidy in the future next generation or any child. Now we will move towards the different phases. Firstly, we will discuss mitotic division, is the mitosis. So we will move towards the different phases of mitotic division. So before beginning the different phases of mitotic division, let's see the diagrammatic representation or a chart representation of these cell division. Is this circle okay? This is the cycle which will represent all the different phases of a cell cycle. As we already discussed, the 95% of a time of a cell cycle is dedicated only to interphase, which is the resting phase. But the resting phase does not indicate that it is the phase where cell rest. No, cell do not rest in the resting phase or interphase. It prepare itself for the next upcoming phases or next for the cell division. So This is the interphase, and this half, which is the 95%, this is the M phase, the mitotic phase, cell cycle of a mitotic division. This is the mitotic phase, which comprises only 5%. Now, interphase, we know there is G1 phase, then S phase, then G2 phase. I'm representing the mitotic phase is quite larger so that we can discuss different points of M phase in this chart. Now, we know the mitotic phase is divided into karyokinesis and the cytokinesis. Here, cytokinesis happens. Cytokinesis is a very small event. This is cytokinesis. This is the karyokinesis. First, the phase is prophase. We all know. And then there is metaphase. Next is anaphase. There is telophase. So we know in what's happened in the G1 phase. In the G1 phase, the growth of a cell occur, increase in the cytoplasm, increase in the cell membrane, increase in the cell membrane means the expansion of a cell membrane, increase in the cell organelle, in some synthesis of a cell, some proteins and RNA synthesis. In the synthesis phase, the duplication of a centriole and replication of a DNA, which is the genetic material of a cell, occurs. Now in the G2 phase, some more growth continues, and here the tubulin synthesis of a tubulin protein is occurred, duplication of some cell organelles like mitochondria and Golgi body. Now, the M phase of a cell cycle is the most dramatic phase. Most dramatic phase means the very large number of event or rapid changes occurring in the M phase. So, M phase is the most dramatic phase of a cell cycle. That's why interphase is known as resting phase, as because there is no much more changes, visible changes occur in this interphase, whereas lots of visible changes will noticed in the mitotic phase. So in the prophase, what happens? Let's discuss this very briefly. In the prophase, what happens? In the prophase, the condensation of a genetic material which is present in the form of a chromatin fiber in our DNA cell. So condensation begins here, and the and the disintegration means nuclear membrane starts to break here, and cell organelles will start to disappear. By the end of prophase, as soon as cell enters in the metaphase, means the end of a prophase and the beginning of metaphase both are same. It means as soon as cell enters in ends the prophase phase, or we may call it in a different way, जैसे ही सेल प्रोफाइल सेल का जो डीएनए है, डीएनए जेनेटिक मटेरियल क्रोमैटिन फाइबर, जो कंडेंसेशन जो स्टार्ट हुआ था उसमें, जैसे ही वो कंडेंस होते चला जाएगा, जैसे ही वो हाईली कंडेंस्ड हो जाएगा, we will say that the cell is in metaphase. These phases are not the boundary which divide the cell that it is in the metaphase or it is in the prophase. The event which are occurring in the cell, जो भी इवेंट सेल में हो रहा है, आफ्टर सीइंग दोज इवेंट, हम उन सब इवेंट को देखने के बाद हम लोग बताते हैं कि सेल किस फेज में है. अगर हमें ऐसा सेल, इफ वी नोटिस अ सेल, हम लोग एक सेल को देखते हैं अंडर द माइक्रोस्कोप और हम लोग देखते हैं कि अगर उसमें लंबे जो क्रोमैटिन फाइबर जो थोड़ा बहुत कंडेंस होना स्टार्ट हो चुका है, तब हम लोग बोलेंगे कि हाँ सेल, ये सेल जो है वो प्रोफेज फेज में. वहां पे कहीं भी हमें वहां पे लिखा हुआ या फिर कोई बाउंड्री लाइन नहीं मिलेगी कि हाँ सेल अब इस फेज में है. ग्रेजुअली एज जैसे ही ग्रोथ ऑलमोस्ट ग्रोथ जो है वो सेल का कंप्लीट होने लगेगा, तो सेल क्या करेगा, अपने आप को अपना जेनेटिक मटेरियल को थोड़ा धीरे धीरे कॉम्पैक्ट करना स्टार्ट कर देगा. तो जब भी हम लोग थोड़ा बहुत कॉम्पैक्शन स्टार्ट देखेंगे, देन वी विल से कि सेल जो है वो प्रोफेज में है. और how we will decide that the prophase is about to end means कि जब सेल न्यूक्लियर मेम्ब्रेन जो है वो पूरा डिसइंटीग्रेट हो चुका है, सेल क्रोमैटिन फाइबर्स जो है वो कंडेंस होकर क्रोमोसोम बन चुके हैं, देन वी विल से कि वो मेटाफेज में. इट मीन मेटाफेज का ये फीचर है कि मेटाफेज में जो क्रोमोसोम है वो हाईली कंडेंस होगा. मेटाफेज का कैरेक्टरिस्टिक फीचर यही है कि मेटाफेज में जो क्रोमोसोम है वो सबसे शॉर्ट हो जाता है, मीन्स वहां पे सबसे ज्यादा हाईली कंडेंस होता है. इफ वी वांट टू ऑब्जर्व अ chromosome of a cell, we will we will not observe only in the cell which are in the metaphase. हमें अगर हम रैंडम सेल्स को अगर नोटिस देखते हैं, तो जिन सेल में हमें क्रोमोसोम दिख रहा है, दैट इंडिकेट्स द सेल इज इन द मेटाफेज. और क्रोमोसोम नहीं दिख रहा है, इट मीन्स वी विल से कि वो किसी और फेज में है, बिकॉज ये सारे फेजेस में विजिबल होते हैं और ये सारे फेजेस में हमें क्रोमोसोम नहीं दिखता है. इन सब फेजेस में हमें वो फाइबर दिखेगा, थ्रेड लाइक अ स्ट्रक्चर का होता है ये. जबकि प्रोफेज में वी विल से कि लंबे लंबे ऐसा क्रोमोसोम टाइप स्ट्रक्चर दिखेंगे. मेटाफेज में सबसे छोटे क्रोमोसोम दिखेंगे, हाईली कंडेंस क्रोमोसोम दिखेंगे. एनाफेज में क्या होता है, वही जो हाईली कंडेंस क्रोमोसोम है वो डिवाइड होने लगता है दो फेज में. और टीलोफेज में हम लोग देखेंगे कि दे आर सेपरेटेड एट टू डिफरेंट पोल ऑफ विद इन अ सिंगल सेल. हम लोग एक ही सेल में देखेंगे कि दो जगह न्यूक्लियस दिख रहा है. जबकि एनाफेज में हमें क्या दिखेगा कि जो मेटाफेज में जो क्रोमोसोम बने थे, जो भी सबसे हाईली कंडेंस क्रोमोसोम थे, वो सारे यहाँ पे आ चुके होते हैं सेल के सेंटर पे, एकदम सेंटर पे एक लाइन सा बन जाता है और वहां पे सारे मेटाफेज में क्रोमोसोम आ जाते हैं. और जबकि एनाफेज में क्या होता है कि वही जो मेटाफेज में जो सेंटर में जो आए थे वो क्रोमोसोम, वो मूविंग अपार्ट, वो स्टार्ट करते हैं एक दूसरे से दूर होना, वो टूट कर बीच से ब्रेक होकर दो डिफरेंट पोल पे जाने के लिए बढ़ रहे होते हैं. और वही टीलोफेज में लोग वही सेम इवेंट देखते हैं कि इसमें जो 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 क्रोमोसोम सेपरेट हो रहे थे एक दूसरे से, वो क्या होते हैं, वो दो डिफरेंट कॉर्नर पे जा पहुंच चुके होते हैं. वहां पे न्यूक्लियर मेम्ब्रेन डेवलप होना स्टार्ट हो चुका हो, डेवलप मीन्स इंटीग्रेट होना स्टार्ट हो चुका होता है. और जो क्रोमोसोम हाईली कंडेंस्ड क्रोमोसोम था वो धीरे धीरे फिर से लूजन अप होने लगा है और क्रोमैटिन फाइबर की तरह स्ट्रक्चर फॉर्म करने लगा है. तो इस तरीके से हमारा कैरियोकाइनेसिस का स्टेप कंप्लीट हुआ है, जिसमें कि जो भी डीएनए मटेरियल था वो पहले क्या हुआ, कंडेंस हुआ, फिर वो मेटाफेज प्लेट पे आया, फिर वहां से सेपरेट हुआ, फिर सेपरेट होके दो डिफरेंट पोल्स पे पहुंच गया. और दो डिफरेंट पोल पे पहुंचने के बाद फिर से वो अपने पुराने फेज में, मीन्स प्रो फेज और जो टीलो फेज है, ये दोनों फेज क्या है, ये दोनों फेज एक दूसरे का एग्जैक्टली सेम है. बेसिकली यहाँ से हम लोग इंटर कर रहे हैं वो फेज, और यहाँ पे हमें एग्जिट करने के लिए ये दोनों फेज में सारे ऑलमोस्ट सेम इवेंट होते हैं, अपोजिट टू ईच अदर. यहाँ पे जैसे कि डिसइंटीग्रेट होता है, यहाँ पे इंटीग्रेट होते हैं, यहाँ पे कंडेंसेशन स्टार्ट होता है, यहाँ पे अनकंडेंसेशन होता है, यहाँ पे कि जो कंडेंस है वो डिकंडेंस हो रहा है. और इन द साइटोकाइनेसिस, जस्ट साइटोकाइनेसिस को हम लोग दो पार्ट में, टू इक्वल हाफ में डिवाइड करते हैं. तो दिस इज द सेल साइकिल. फ्रॉम सेल here enter, here two daughter cell will form, cell enter here, the growth occur, cell will grow. Now here the only, no, represented in the form of DNA. This is cell membrane, this is neutral membrane. Here cytoplasmic content will increases. Now what happens here? One single set of one pair of chromosome is there. यार सिंथेसिस में क्या हो रहा है यार, जैसे कि मैं 2 डीएनए स्ट्रैंड यहाँ पर बना रही हूँ, सिंथेसिस में क्या हो रहा है कि ये जो 2 डीएनए स्ट्रैंड है वो क्या हो रहा है, हमारा रेप्लिकेट होकर जैसे कि ये 1 स्ट्रैंड था तो इसका साइड में ही दूसरा स्ट्रैंड बन गया, और इसमें भी देखो ये एक जैसा कि हमारा एक डीएनए स्ट्रैंड था तो ये रेप्लिकेट होगा और यहाँ पे दूसरा स्ट्रैंड बन जाएगा। फिर G2 फेज में क्या होगा, ये G2 फेज में क्या होगा कि हमारा सेल मेम्ब्रेन तो वही रहेगा, न्यूक्लियर मेम्ब्रेन भी वही रहेगा, जेनेटिक मटेरियल भी वही रहेगा, इधर कोई चेंजेस नहीं आएंगे। और जो बाहर का जो सेल ऑर्गेनिल्स है वो सब अपने आप को ग्रो करेंगे, अपने आप को रेप्लिकेट करेंगे, सब कुछ प्रोटीन का सिंथेसिस होगा। सो हियर let's talk about the ploidy and DNA content of the cell here, as we haven't discussed diploidy and DNA content earlier, so we will discuss in this table. Here in the G1 phase, the DNA, see, we are saying that there is a 2 chromosome, means 1 pair of a chromosome. There is 2 chromosome means 1 pair of a chromosome. 1 pair of a chromosome means represent it is a diploid. It is the diploid. 2n represent for diploid. हमें डिप्लॉइडी को इंडिकेट करता है। डिप्लॉइडी को इंडिकेट, ये जो 2 डाई मीन्स 2, तो ये 2 उसको करता है, और ये जो n है वो हमें क्या नंबर ऑफ अ क्रोमोसोम। As we know now, and that human cells have 23 pair of chromosome. So I am representing only 1 pair of a chromosome here, whereas in the human cell there the 1 pair here, 1 pair, there is for 23 pair. It means 46 chromosome in our human body, but I am representing only 1 pair of chromosome here. So here ploidy is 2n, number of a chromosome is 1, and ploidy is 2n, means diploid. So what is the DNA content here? Here the DNA content is, if we are saying 2 DNA, 2 strands of DNA, then we will say the content is 2C. C is just representing the 1 chromosome. Okay, here is 2C. 1 chromosome means 1 strand of a DNA. Here we see diploidy is still same, as because there is only 2 centrome. Will not say centrome here. Here ploidy will remain same because the cell is not divided at, not multiply. There is no multiplication of a cell. There is no division of a cell. The cell is remain same. Only the thing happened in this is that the single strand converted into 2 strands. We copied the 1 strand and we copied the 1 strand so that we get the 2 strands which are exactly same and similar. So here the ploidy remain again 2n, but the DNA content, as because earlier we had 2 strands, and here we have 4 strands. So DNA content is 4C, and the ploidy is still diploid, which is 2n, and here all the things will remain same. Now in this phase, there is there is no change in prophase, nothing happened in the metaphase. These the genetic material all remain same. Here in the anaphase, not permanently, as because mitosis is M phase is itself a very short period, in which anaphase is the shortest phase and prophase is the longest phase. Anaphase is the shortest phase. In the anaphase, the single chromosome, which is the two strands of a chromatin thread or DNA, the two thread which are joined, which are the two threads of a DNA which are joined in the prophase, metaphase. It remains joined in these two phases. While in the anaphase, these two joint strands get separated, and start moving towards the two different sides. If they are arranged in the here we imagine that it is a metaphasic plate. Here the two strands, both the strands which are joined here, start separating from each other and start moving towards the different pole. They both are moving opposite to each other. So here the DNA content is remained 4C, but the ploidy of a DNA will become 4C, 4N, which is very temporary. I'm telling you all this just for extra knowledge, just to understand what are the events occurring in the anaphase. Here in the anaphase, this is the temporarily occurring. In the anaphase, the number of DNA strands remains same. The thing happened in this is the both strand of a DNA, same strand of a DNA joined by a centromere. This is known as a single chromosome. But as they divide into two different, they both are known as one chromosome, and this is a single chromosome. They both became two different chromosomes. That's why the number of a chromosome here, which is N, became 4 times, 4N. And here is 4C. Will not say 4C. Here the nucleus of a cell divided into two. This will remain in same in the telophase. In the cytokinesis, this 4C, 4N divided into two equal halves and give us 4N divided, 4C also get divided, and we got two cells of 2N and 2C. We got two cells of 2N and 2C, which is same as this G1 phase. Okay, got it? These are the here we discuss about the ploidy and the amount of DNA content in a mitotic division. Let's see the different phases of mitotic division, which is the prophase, metaphase, anaphase and telophase. As we already discussed it very detailed, we will write on the points and try to represent you diagrammatically.